Dear students, so in this lecture we are going to explore environmental genomics. So we do know now the basic tenet of microbiology is that the DNA stores the essential information for cell, and then it's transcribed into an RNA, messenger RNA, which then is taken to certain parts of the cell, and it is used as a template for making proteins, which is translation or expression of the gene. So DNA stores the information, and then it undergoes the cell undergoes various steps so that a protein corresponding to the DNA can be made. Now, if I want to understand the functional characteristics of the cell, if I want to understand the behavior and the preferences of the cell, then I need to know what its DNA is. So genomics is when I can actually look at all the genes of a cell and then predict the behavior of the cell. So think of it this way: if you know the code of the computer, if you know the software code which was written by some computer algorithmist and scientist, if you know the code, you can know what kind of work your computer can do, what kind of behavior to expect from the computer. Same is with the cell. If with microbes we can know their genomes, we can guess, we can make a better guess at what functions and expressions are to be expected from the microbes. So when we apply this technology to environmental systems, environmental microbial communities, we call it environmental genomics. So that's what we'll be exploring this week, and let's see what we have here today. So it all boils down into sequencing the DNA. So once I know the sequence of DNA, A, T, G, whatever it is, I can understand what amino acids it would code for, because A, T, G, for example, would be transcribed into an RNA where T would be replaced by U, and then this RNA would go to where the proteins are going to be made, and then the protein ATG, whatever this codon codes for, that particular amino acid will be attached to the amino acid chain. So if I know the sequence of genes, or if I know the sequence of DNA, I can know the sequence of amino acids, and if I know the sequence of amino acids, I can do in silico modeling and understand what its secondary structure would be, what its tertiary structure would be, what its quaternary structure would be, and thus what its behavior would be. And usually what we have done in past few decades is that we have grown microbes in lab, we have tested them under different conditions. So we know, for example, this microbe will eat glucose but not lactose, or we know this microbe is resistant to some carbapenem antibiotic but not resistant to something else. So when we know these behaviors, we sequence the genome, and we can say, okay, if a microbe is perfectly similar to this, if the genome of another microbe is perfectly similar to the genome of the one we grew in the lab, we know that they are perhaps same microbe, or they are very closely related to each other, and thus we can also predict, without even growing the other microbe from the environment, we can predict its behavior. For example, if I have a methicillin resistant Staphylococcus aureus in my lab that I am growing, MRSA, and I take an environmental sample, and I know that there is only one bacteria, let's say, and I sequence it, the entire genome, and then I match it with the genome of the microbe that I have in my lab, the MRSA in my lab, and if I see that they are 98% similar to each other, very highly similar to each other, I can guess that maybe the microbe that I have isolated from the environment is also methicillin resistant Staphylococcus aureus or MRSA, or it's very closely related to each other. So just by looking at the genetic typing, I can say that maybe there's a very good chance that my environmental isolate is also resistant to methicillin. But how do I sequence? That's a major question. Now, if you remember, the DNA molecules are very, very small, and they're very tightly coiled to each other. They make a very beautiful double helix which is very tightly coiled to each other. It's called supercoiling. Now, sequencing means not only do I need to look at them nucleotide per nucleotide, but I also need to have a mechanism. When I am reading nucleotide by nucleotide, a signal is created which can be read by my instrument, which can be translated into meaningful data for me. But also it should be done at rapid enough pace that it makes sense for me. Otherwise I might have to wait months and months to get whole genome of a single bacteria. So the first groundbreaking work was done by Frederick Sanger. Here's a picture of Frederick Sanger on the left panel, and when he developed his Sanger diadoxy method for sequencing genes, he actually got Nobel Prize for it. And even though this is the first generation DNA sequencing, by the way, even though now we have second generation, third generation and fourth generation DNA sequencing techniques, the Sanger sequencing is still used for many processes because it is so helpful. One of the advantages of Sanger sequencing is that you can sequence really long fragments of DNA. The other thing is the DNA sequencing technique that Frederick Sanger invented. It was based on three major principles, and those principles are still used by many sequencing technologies right now. So the first principle that Sanger worked on, proposed and used for his Sanger sequencing was using the DNA syntheses. So if I want to read the sequence of something, the intuitive approach is to break it down one by one, break down a nucleotide. Okay, this is ATP, so A. This is GTP, so G. This is CTP, so C. This is one approach to go, but he said no, not by breaking down, but by syntheses. So I have a template of, like I have a DNA, I denature it, now it is single stranded, and now I have a DNA Taq polymerase which wants to make its replica, so more complementary strand. So I put DATP and I say okay, A attached, so the other side should have been T. Okay, G attached, so it should have been C. C attached, so it should have been G. So by synthesis, by seeing what is being attached when I am making a complementary strand, I can do DNA sequencing. That is the major philosophy of Sanger sequencing. This next beautiful thing that he did was he used DDNTP, dideoxynucleic acids. Now dideoxynucleic acids, they don't have an OH radical in the third position, but they have an H. So when they have an H, not an OH, they cannot continue forming chain once something is added. So let's say I have, it would look something like this. Okay, so let's say this is a part of the DNA that I have, and it is single stranded DNA, part of single stranded DNA, and let's say this is the direction at which I am sequencing my DNA. Now remember, the first underlying principle of Frederick Sanger sequencing technique was sequence it by DNA synthesis. So I might have my Taq polymerase here, and it wants to do it by synthesis. So now let's say these have already been paired up. Now here I have C. So when I add, now here ideally it should pair up with G, right? It will form a very strong triple hydrogen bond with G. So instead of adding DGDP, I add dideoxy GDP, so it has one OH radical less. So when DDGDP comes here and attaches here, it cannot, because it has dideoxy, instead of OH it has H in third position, it cannot form a further link with the G that wants to attach here. So the reaction stops here. So the reaction will stop here. So this is how Sanger sequencing works at every step. So now the reaction has stopped here. Okay, we know that G will come. So now we have this A, D, C, G. So we divided it into 4 wells, and I will show you how we do that. We divide it into 4 wells. Now we already have ATCG. There is room for another G to attach. So when in the well that I put DGTP again, the G will attach here again, right? So now the reaction will terminate. So at every step the reaction terminates. So I need to have many wells in which this reaction is carried out. Earlier this reaction was very painstakingly slow because there was a lot of manual work to do, but now we do Sanger sequencing in automatic robotic instruments. So this is relatively much faster. The other thing is, every time the G attaches here, initially when Fred Sanger worked on it, they were radioactive labeled. So we could catch the radioactive signal, and we knew, okay, the G they responded, a T it responded, or A it responded, or C it responded, and we could create a sequence for this single stranded DNA. But now we don't use radioactive material anymore, we use fluorescence instead. So every time some dideoxy nucleotide comes and attaches to it, a fluorescent signal is released, and this is read by our detector. So the detector then tells us ATGC and so on and so forth. Okay, so the three principles of Fred Sanger sequencing, Sanger diadoxy method for sequencing are: use DNA synthesis method instead of DNA breaking method, use DDNTP to block chain extension. So every time nucleotide is added, the reaction terminates right there because we are using dideoxynucleotides, and use labeled precursors for detection. And as I mentioned, in times of Fred Sanger, we used to use radioactively labeled precursors, but now we use fluorescent ones. So this is a picture showing how the sequencing happens. So I have this sequence here, and we divide it into 4 wells, and in each well we add a particular dideoxy nucleic acid. So I add DDATP in 1, DDTTP in 1, DDCTP in 1, and DDGTP in 1. So if I get a signal from the first one, I'll call it A signal, second I'll call it T signal, third C signal, fourth G signal, and so on and so forth. So remember, extension happens 5' to 3', and then when A is added, I get a signal here. When T is added, I get a signal here, and so on and so forth. And then I can read my signal. Okay, first A was added, then T was added, then T, G, A, C, G, C, A, T, and so on and so forth. And I can know what my read sequence is, and I can know what my complementary sequence is. And this is how the sequencing happens. Now, the way it works is, my dear, is that let's say this is the fragment that I want to sequence: GAA, CT, T, GCT, GT. So this is what I want to sequence. This we attach it to a primer. These primers are short oligonucleotides. Oligo means small, so they are short length nucleotides polymer, and they are in lab attached to the sequence, the gene that or the DNA element that I want to sequence, and these are labeled. Earlier they used to be radioactively labeled, now they are fluorescent labeled. And when the reaction completes here or does not complete here, I know what has happened. So I add DDATP, it terminated here, I read an A. So A is read. Next step, again A is read. Next step, here C would be read. Next step we have T, so T would be read here. And then T would be read here. Then we have a G, so G would be read here. Then we have a C, so C would be read here. And then we have a G, so G would be read here. And then we have a read sequence ready. So it actually looks like this. We have nowadays what we do is, instead of breaking it into 4 different wells and doing it painstakingly slow, so this is a small fragment of what it looks like, but we have pictures, you can Google them, I encourage you to look it up. Pictures of Fred Sanger reading his electrophoresis sheets, and they are very long sheets of these blots, and then you have to read, okay, A, T, A, G, C, very long time to write it down. But now we have a very wonderful method. We run them through a capillary tube, so it's like electrophoresis, capillary electrophoresis, and they are sequenced with di terminators. So when we have A, and then this reaction stopped at A, this reaction stopped at T, at G, C, and so on and so forth. And when they pass through the capillary tube, there is an excitation laser, and it excites these fluorescent tags, the terminator tags, and as they pass we get a signal. So when we get signal of 2 intensity, like twice the green signal, we don't get green 1, green 2, we get twice the intensity of A, of green. So we know that green stands for A here, so we have 2A. If we get thrice, we know there are 3 A's. Okay, now this is called the first generation sequencing method. So first generation DNA sequencing. Now coming to second generation DNA sequencing. In second generation DNA sequencing, the major advancement was that we could multiplex these sequences. So here we are sequencing a single strand at one go. So it's very slow. very slow, but in second generation we could multiplex them. So we found ways to make micro tubes, we found ways to make micro wells. In each well we could put one fragment of DNA, and then we could sequence it, and then the robotic arm could read from each well and know what the sequence is. So this was developed in the second generation sequencing. Now the advantages of second generation sequencing is that it was the first time, and it actually is very near past, not very long time ago. In near past, the second generation sequencing techniques have highly reduced the price of sequencing per base pair. So now we can get earlier we had to spend lot of money to get Sanger sequencing, but now we get very rapid and very cheap sequences per base pair by second generation sequencing. There are two basic platforms in which we use for second generation sequencing. One is immersion PCR, the other is bridge PCR. So let's talk about both of them, but let's start with the first step. The first step is library preparation. So in library preparation, whatever DNA that you want to sequence, now this could be whole genome. It doesn't have to be a part of DNA or a particular gene, it could be whole genome. And the first step is that we shotgun this DNA, so we break it into small fragments. Now each of these fragments are then individually sequenced, and we usually, when we prepare the library, when we break them apart, we add certain primers, adapters, markers that tell us, okay, this is the right size, this is what I want to do, and when I sequence I know what I am sequencing. So this is also called shotgun sequencing, because we basically shotgun the whole genome, and then we take the right size of amplicon and we sequence that. So step 1 is library preparation. This is usually done in vitro. So we fragment the DNA, so the DNA we fragment it, so the blue one is the fragmented one. Then we add adapters, and in this picture we are adding two adapters, green and red. They both are important because they both will tell us, okay, when I detect A, when the sequencer detects A, it knows, okay, these are the readings, the following readings I have to take. When it detects B, it knows, okay, the reading before is what I have to read. So this is in vitro ligation. There are many ways of doing it. One is amplicon, the other is shotgun, the other is cDNA techniques of preparing library. Now amplicon sequencing would look like this, that we take these fragments and then we clone them in different E. coli, and then so we have created multiple copies, we have created amplicon, multiple copies of each of these fragments. Now when each of these fragments have been amplified, I can then sequence them individually. In shotgun, as I mentioned, we just break it into fragments, we ligate the adapter, and we are ready to sequence. Now the second step is clonal amplification. We can either do it in clones, or we can do immersion PCR and bridge PCR. They are usually faster than clonal amplification, because clones take time to grow and you have to pick so many colonies. In immersion PCR is like a general PCR, but instead of having a single well where the PCR reaction takes place, we create micro oil emulsions. So these are micro emulsions of oil, and each of these emulsions, they carry all the essential ingredients for PCR. So they have primers, they have the DNTPs, they have the gene fragment that we want to sequence, and then at the end of immersion PCR, each of this emulsion that had the starting DNA material will have multiple copies of your portion of DNA that you want to sequence. Now usually immersion PCR will carry beads in them. So the way it works is that we have a bead. So the way it works is that in immersion PCR we have beads. Now this bead will, a part of your DNA that you want to sequence will attach to this bead. It will be part of your emulsion, micro emulsion, which will have the primers you want, will have all the DNTPs you want, the labels you want, and then it will undergo reaction, and when the reaction converts, you will notice that you will have multiple copies of this particular DNA fragment. And then in the next step you can separate these beads and the sequencer can read them. And because it reads them multiple times, because there are multiple copies, it can ensure the accuracy of what you are reading. In bridge PCR, instead of doing emulsion PCR, what we have is we have a bridge, a plate. Now the adapters, they attach to one end, they attach to the bridge, to the plate, and the other end also attaches, and then the DNA tag will come and sequence them. So it looks something like this. So this is the plate you have. So one adapter end attaches here, and this is your DNA you want to sequence. The other adapter end attaches here. The DNA tag will come, it will create a this is single stranded, now it is double stranded. So it has sequenced it as it synthesized it, and in the end what you will have is, once it comes here, this will be released and you have a long sequence chain here, which, and you can create multiple copies. So you have multiple adapters here ready to attach to DNA fragments. So this is bridge PCR. The emulsion PCR are used by the ion proton sequencing technique, by 454 Pyro Sequencing, and by Illumina sequencing. And I think I will be briefly going through them to tell you what are the differences in Pyro Sequencing, semiconductor sequencing, and fluorescent nucleotide based Illumina sequencing. So let us look at Pyro Sequencing. Pyro Sequencing, first step is library preparation, and after library has been prepared, we do emulsion based sample PCR. So first we had the genetic material, we split it into small pieces. Now they were converted into single stranded, and look, they have blue and purple adapters. So these libraries will then be distributed in emulsion. So these are emulsion, each emulsion has bead and a genetic fragment. Then they undergo PCR amplification, and when they undergo PCR amplification, they will create another strand of genetic material. Then they dissociate with each other, and now we have two strands. You see how the dissociation happens in denaturing step, and at the end of it we will have several thousand copies of this genetic material sticking with the bead. Now each of these bead is then robotically put into a micro well. So these are millions of wells. So here we have on average 1.6 million wells. So they are put in millions of wells where they are sequenced. Alrighty. Now the way the technology of Pyro Sequencing works this way. So once they go into these wells, you have your DNA, and now another step of amplification will happen. When this step of amplification happens, when the DNTP attaches, so again this is Sanger, it uses the same DNA synthesis principle of Sanger sequencing. When it attaches, let us say DTP is attaching, when T attaches, it releases a pyrophosphate. This pyrophosphate is consumed by AMP to make ATP, and when ATP is formed, ATP is consumed by luciferase, which is an enzyme. When this enzyme comes in touch with ATP, it consumes ATP and releases light, and this light is seen. So let us say in this well I add G, but I do not see any light. So I know that no pyrophosphate was released, hence G was not accepted. So then I will add T. When I add T, I notice that I noticed light, I observed light, so I know that pyrophosphate was released, hence T was accepted. So I know that now the nucleotide that just has been added is T, thus in the template I have A. So now this way I have sequence. This is Pyro Sequencing. Obviously you can guess the name is derived from pyrophosphate that is released every time a nucleotide is added. So basically we have DNA, we have DNTP polymerase, and it releases pyrophosphate. Pyrophosphate attaches with AMP and PEP, forms ATP and pyruvate, and this ATP attaches with luciferin, it gets oxidized in presence of luciferase enzyme and releases light. Now luciferase enzyme is the same enzyme that allows fireflies and many deep sea microbes and deep sea mammals to release light. So this is Pyro Sequencing for you. Now this is really neat. To summarize, we prepare the library, we have emulsion PCR. Now at the end of emulsion PCR, at the beginning of emulsion PCR, ideally in each emulsion we have all the things we need for PCR. We have a bead, and there is one genetic strand attached to the bead. With multiple steps, we have several thousands of the same genetic element, now replica of these attached to the bead. Each bead goes to a microvalve, and in this way we generate millions of sequences, because there are 1.6 million valves. So we will generate millions of sequences per run. Okay. So here you notice that DNTPs are, now this is slightly different. Now this I think is part of the third generation sequencing, because if you notice here in Pyro Sequencing we are relying on optical measurement, we are relying on how we can measure the light. So optical cameras which are so tuned to such small amount of light generated, Usually, by the way, this is not so tiny amount of light, because remember we have several thousand copies per bead. So that when we add DTP, for example, in this case, each of these would ideally release some light. So collectively we will get a considerable signal. But to have such a fine camera and such a small optical recorder at such a for such high throughput sequencing is very expensive. So the third generation sequencing techniques are the ones that are past the optical detectors. Now they use they use electrochemical detectors. So this is DNTPs are passing in sequence as the sequencing is happening. The pH changes. You know, a proton is released. That proton is detected. So if I pass DTPs, so I know the T was accepted if a proton is released. So in fact, ion torrent sequencing, which uses this third generation sequencing method, is they dub their sensor as the world's smallest pH meter. So proton is very easy to detect, pH is very easy to detect compared to the optical signal. This is the advantage of the third generation sequencing technique, and ion torrent is one of them. I'm actually looking forward to working with some ion torrent sequencing in near future. So by the time you read this lecture, you hear this lecture, I have probably already worked with ion torrent sequencing. So when the pH is measured, we know that following nucleotide has been accepted. And okay, here is how it works. Let's read through this because it's really well written. So a proton is released when right DNTP flows by and is incorporated into the complementary strand. This proton release changes the pH, because remember pH is minus log concentration of H, which changes the charge in the ion sensing plate. So ion torrent, so we have this ion sensing plate and it changes the charge, which creates a voltage across the ion sensitive transistor that sends a signal to computer to record the DNTP species. Again, this is very high throughput. This is third generation sequencing. So remember, first generation sequencing we have Sanger sequencing. Second generation sequencing we have multiplexed it. So we have high amount. We are creating high amount of base pair data for very short small amount of cost in very little time. Third generation sequencing, we are past the optical measurements and we are looking at the electron measurements or proton measurements. So this electrochemical sensing make things cheaper. So ion torrent sequencing is much more cheaper than optical based sequencing techniques. In fourth generation sequencing, and this is the example of nanopore sequencing, it's very different. In nanopore sequencing, for example, we have this protein across a membrane. So this is a transmembrane protein, and what happens is that the DNA double helix is in a way denatured, uncoiled, and one of the single strand passes through the protein. And as it passes through the protein, in fact it is excreted through the protein. When it passes, it changes the electrical signal, and the signal depends on what is passing through it, right? So let's read through this so that you understand this more clearly. And when the signal is captured, we know what has passed through. The advantage of nanopore sequencing and fourth generation sequencing is that we can generate data for longer genetic fragments. For example, ion torrent, it does really good job around 200 to 300 base pair. Pyro sequencing will go to 400 to 500, maybe even 600 base pair. Sanger sequencing can go from anywhere from 500 to 2000 base pair. But the nanopore sequencing can do a much better job. So this example is of, by the way, Oxford nanopore based sequencing. So DNA can be sequenced by threading it through a microscopic pore in a membrane. Bases are identified by the way they affect ions flowing through the pore from one side of the membrane to other. So we have DNA double helix uncoiled passing through here. So one protein will unzip the DNA helix into two strands. It will reject one strand and pull the other strand in. The second protein will create a pore in the membrane and it holds an adapter molecule. Now the ions are also flowing through this. As the ions are flowing through this, a current happens. So current is basically flow of electrons or flow of ions, and each base as it passes will block the flow. Now A, T, G, C, all of them block the flow to different degrees. By looking at this impedance, this blockage in flow, we know what was present. So if you look at this spectrograph here, we notice T has the highest block, G blocks this much. So every time I notice this amount of blocking, I notice G. Every time I notice this amount of blocking, I notice T. And what adapter molecule does is it keeps the bases, it checks their speed of secretion or excretion. so it makes sure they pass slowly enough that we get a signal. This is nanopore sequencing, and last 2 years the company has done a very good job trying to promote it. And the beauty of 4th generation sequencing is how portable the instruments are. So iron torrent, for example, it requires an air conditioned room, it requires to sit on the right bench, and it needs to be maintained in the right way. However, nanopore sequencing is size of a flashlight. So you have a flashlight, you just carry nanopore sequencing with you, put your sample here, and it can be so fast, you can sequence the entire human genome in few hours. So this is nanopore sequencing for you, and there are 3 different technologies right now in market. One is the Oxford nanopore technology, whose example I was showing you here. Then there is Genia technology, and there is Stratos genomics. So in all of them use semiconductivity based measurements to know how current is flowing through the pore, and then they translate that into DNA sequence. But there are slight differences in each of them. Okay. Now once we have generated the sequence, the next challenge is how are we going to analyze the sequence. Okay, I have a long sequence A, T, G, C, G, C, T, 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 A, whatever, but I don't know how to make sense out of it. So what is the difference between G, C, A, T, T, C, G, A, G, C, T? Is it even meaningful data, or is it just noise or just meaningless data? Because we notice that in a cell, in its DNA, it carries informational genetic codes and it carries non-informational genetic codes. And just by looking at it, there is no way for us to know whether it's informational or non-informational. So for example, remember I talked about start codon and stop codon. So between start codon and stop codon, ideally we have an ORF, open reading frame. So we have genetic material, informational material. And near start codon there should be a ribosomal attachment site, binding site. So in the ribosome will attach there, and then it will allow translation of the mRNA that will be formed by this ORF. But between multiple ORFs there are still sequences, and they do not code for anything. They are nonsense in this way. So how do we tell them? We tell them by feeding the sequence data that we have generated here, these, and typically they are in form of FASTQ file or FASTA file. So the sequence that we'll generate either from the first generation Sanger sequencing, second generation Illumina sequencing, third generation iron torrent sequencing, or fourth generation nanopore sequencing would look like this. And so on and so forth. Right, and the file, the output file that you will get will either be in the form of FASTA file, so .FASTA or .FASTQ. The difference between FASTQ and FASTA file is that the FASTQ also carries the quality information. So not only will it know AT, GC, TTA and so on and so forth the information, but it will also have quality scores for each of the base pair. So if the quality score is less, then I can ignore this, saying that this is poor quality, this might just be a chimera, or this might be a misread. Let's reject this sequence. So most of the sequences will give you FASTQ file, and then you can put FASTQ file in your computer, and you can ask your computer to eliminate the poor quality reads, and then you are left with a good quality FASTQ file. Now in order to analyze, the computer does not need quality scores. Once you have already eliminated poor quality score nucleotides and reads, you do not need the quality information. So most programmers will convert this into FASTA file. Now let's take a look how your typical FASTA file will look. A typical FASTA file will have a mark like this. It will have the name of this read. So it could be A, B, C, 1, 2, 3, 0, 4. Something like this, and then in the next line you will have the sequence. It might be many lines of sequence, and then when the sequence ends, for example, you will have another line, and you will have, and then in next line you have your more sequences. So in one of the homeworks for this week, what you will have is you will get a FASTA file, and I'll ask you to annotate it. So this is your FASTA file, and in fact, and another homework that I am giving you is I'll give you 2-3 different sequences, and I'll ask you to create a FASTA file. So even if I have only one sequence, let's say Sanger sequencing I am doing and I only have one sequence, this is still a FASTA file, a standalone FASTA file. I can combine two FASTA file by just writing the components of the second FASTA file below, then in the next line here. The next line after where the sequence of the first FASTA file ended. So I can combine two FASTA files. Now once the computer gets a FASTA file, it needs to identify what are called as ORFs, open reading frames. So once it has identified open reading frames, only then it can go ahead and try to make sense out of the sequence. Now how does it identify open reading frames? Let's take a look. Now in order to identify open reading frame, the computer undergoes a particular algorithm, and it looks like this. Computer finds possible start codons, then computer finds possible stop codons, then computer counts codons between start and stop codons. Then computer finds possible ribosomal binding sites. It calculates the codon bias in the putative ORF, tentative ORF, and then it decides if the ORF is genuine or not, and then it creates a list of probable ORFs. So let's take a look. So let's say this is one of the sequence, and I have left the places where I don't want to write the sequence in blank. So the first step for computer would be, it will go through all the sequences and look for start codon. There is only one start codon that microbes use, and that is AUG. So the computer will go through and it will look for all ATGs, all AUGs, and when it has identified the start codons, the next step it will do is, it will look if there is a stop codon after the start codon. So it will search for stop codons. Now there are three stop codons, UGA, UAA and UAG. So you know why it went for start codons first, because this will minimize the number of sequences that it needs to analyze. So then it will look for stop codons. So the second step is stop codon. Once it has identified the stop codon, and you know there might be multiple stop codons, let's say UAA is here, UAG is here. So there are two stop codons after the start codon in this particular sequence. So the computer identified two stop codons after the start codon. The third step it does is, it calculates how many base pairs are between start and stop codon. Let's say this is 20 base pair. Okay, and let's say this is 600 base pair. Which is more likely to be the actual ORF, the actual open reading frame that carries information. 20 base pair, there is no gene, no protein that is so small. So very less likely that this will be an actual ORF. So the computer will reject this as a stop codon. It will reject this, that this is not a right stop codon. Either there is a sequencing error or it's just not a right ORF. Now if it looks at UAA, this is 600 base pairs far away, so there are some 200 amino acids, which is a decent size for a protein, 200 amino acid protein, and thus it will say, okay, this might be my right stop codon. Okay. In the next step, what computer does, once it has found a start codon, it has found the right stop codon, it has confirmed that okay, the base pairs between start and stop make sense, it will look for ribosomal binding site. So it has a particular sequence, and this sequence, if it matches the sequence for ribosomal binding, where the ribosome can come and sit and bind, ribosome also has some sequences, then it will say, okay, I have identified an ORF. The next thing what the computer does before it confirms that this is an ORF. So see, computer has a lot of data, millions of reads, and it's trying to reduce the number of data to meaningful data, sensible data. So first, those who don't have start codons, reject them. Those who don't have start and stop both, reject them. Those who don't have stop at the right position, reject them. Those who have start and stop and at the right positions with the right amount of base pairs in between but don't have RBS, reject them. Those who have all of them, let's find out their codon bias. So in biology we have remember codons? So three nucleotides together, they code for an amino acid. This is a codon. So there are some codons that code for a singular amino acid. So because we have 4 different kinds of nucleotides, and codons are based on 3 nucleotides. So in first position we can have 4, second we can have 4, third we can have 4. So basically we can have 4 to power 3 codons, 4 to power 3 codons, so 64 codons. But we only have 20 some amino acids. So you can say on average each amino acid is coded by 3 codons. So but each bacteria, each microbe has a preference. I like to use this particular codon for coding for this amino acid, not the other 2, or not the other 3 or other 4. So this is codon bias, they have a codon preference. So what this computer will do is then it will find out what codons am I reading in pairs of 3, and are these the preferred codons of rest of the microbe. It will compare with other sequences it has got from this microbe or not. If it is very different, then it will reject this, because the microbial preference for codon, or the codon bias in a microbe, is specific to the entire microbe. Now in some cases, now please note this very clearly, in some cases it is possible that this is an actual ORF, it actually codes for sensible data and is expressed into a protein, but it has a distinct codon bias from rest of the genome. In that case, the computer can say maybe it is possible that this particular gene, this particular component of genetic element was not originally of the microbe, but the microbe incorporated it from its environment or from other microbes, other bacteria, other protozoa. So this is horizontal gene transfer, and we will talk about it soon. So this is how a computer decides structure of an ORF. And the next step here is, now I have the ORF, this is my unknown genome by the way, and the next generation sequencing machine made small fragments of it, then identified the right ORFs, and once it has identified the right ORFs, this is what I am left with. And then we have genome assembly software. So these are assemblers. These assemblers will, what they will do is they will align these with each other. So all the ORFs are aligned to each other, and whenever there are significant overlaps, we say okay, yeah, look, they are overlaps, so these are overhangs. So because they are overhangs, they were initially part of the same genetic material, but then it was split from different places and then sequenced. So it will reconstruct genome. Often we cannot reconstruct the entire genome, but we can convert these sequences into scaffolds, which are longer strands and the more meaningful thus. And at times, very rarely, we can also reconstruct the whole genome, but reconstructing the whole genome is much more expensive, because there are gaps that are left, and filling these gaps is very intensive and expensive process. So dear students, this is all for today. In the next class, we will go ahead and we will explore more about environmental genomics, more about the tools that are used for environmental genomics, and and I must mention that this is still a growing field right now. In fact, it's one of the latest things. It's called as bioinformatics, making sense of the information from biology, and in the next class we will talk about them. That's all for today. Thank you.